Okay, so this is the last lecture for Psych 3280, Neural-Based Subconsciousness. And today I'm going to talk about frontiers in consciousness research. Okay, so just to start, to recap the week 11, in week 11, I outlined integrated information theory of consciousness, which starts with a proposal of the level of consciousness, and then there is a boundary of consciousness, and then within that, how integrated information composes, determines the contents of consciousness for the humans. And I introduced it quickly, how to compute the integrated information, and also the complex, which corresponds to the border of consciousness, in using a very simple situation model. And then I also, towards the end, explained how IIT can explain consciousness in patients without cerebellum, why there is no effect on the consciousness, and also split brain. Why split brain patients seems to have two consciousnesses within one body or one skull, and then conjoined twins who can potentially have two consciousnesses, and then with a shared input or thoughts possibly. All right, so this week I'm going to outline the frontiers in consciousness research, and with some exemplary research from my own lab. And then also lists empirical questions with strategies and frameworks and techniques that can be addressed. And then also situate the consciousness research in a broader context to wrap up the course as it's at the conclusion. The learning objectives of this week is to be able to answer the questions such as what does IIT propose about the Qualia? This is a sort of the last missing part of the IIT. And then how can we characterize Qualia structures? So the structures are the key concept in this week's lecture. And then I also will address the question of what are the possible empirical experiments that can be used to reveal the relationship between the Qualia and the brain structures. And then finally, what are the societal impacts of consciousness research? And then I added my recent paper with Dr. Alex Meyer, and which outlines some of the future perspective of the consciousness research as an optional reading. Okay, so just to get started, I start from the question from week 11, but actually this has been the sort of the remaining question for over the last 5-6 weeks actually, which is that how does IIT explain contents of consciousness? And for short, I will use the term Qualia. Basically, what Qualia means is the contents of what you are seeing, like redness of the red, or shape of the object, or face, whose identity, or whatever you are conscious of. That's what I call Qualia. And IIT explains level of consciousness as a big phi, or the set of the integrated information that is lost at the level of a system. How does it actually get collapsed by cutting by introducing the minimum information cuts? And the border is corresponds to complex in IIT, which is a local maximum of the subset of the physical substrates that maximize this big phi. And then the Qualia, or contents of consciousness, corresponds to a connected pattern of the small phis. So each of the components can generate integrated information, as you if you recall from the last week. And then how does it actually get connected and then form the pattern that corresponds to exactly what you are experiencing at each moment? That's what IIT tries to explain or proposes. And first, I'm going to talk about this, Qualia or structure according to IIT. So the fundamental, just to remind you, the fundamental questions in consciousness research has been that, the correspondence between what you consciously see, and what your brain is doing. And, in particular, a subset of the neurons, neuronal activity. How does it correspond to, qualia, basic qualia of the dog? And this link between mind and matters is the fundamental questions in consciousness research. And some people call it as a heart problem. Some people call it as different names. And we have lots of clues about the link. This is the theme that I've been trying to establish over the last, weeks. So one of the example I have emphasized is this example of the brain area around, ventral surface of this, human brain. And here we have a cluster of the areas which becomes activated when the stimulus includes some kind of phase. And then you perceive that, oh, there was a phase. And also with, associated identity, emotion, and also gender and so on. They tend to elicit activity on both of the sides, actually. But more important thing is that the right side of the brain tends to have a cluster which can also generate phase percept when it's activated. So these lines are the ones that corresponds to the pairs of the electrodes. When they get stimulated, they elicit the phase percept. On the other hand, next to that, there is a cluster of the areas that prefers color stimulus. Okay, and also when it's paired and then stimulated, they produces a rainbow-like experience. And of course, as a subject who are implanted in this electrodes, they have no idea which one is selected and then stimulated each time. So this is the best probably we can get to in terms of the causal explanation between the qualia and the brain activation. Okay, so but the key for us is that it's not really exactly where this kind of thing happens or the existence of something like this happens. The critical question is why stimulating these areas elicit the qualia of color and face. Because if you don't know anything about the brain areas, then they look really homogeneous. And if you use a microscope and then to look at the details of the neurons that are there, or if you use a DNA analysis, most likely you don't find anything different between this red part and the blue part. But somehow there is something that goes on within this red areas and the blue areas that eventually gives rise to difference in quality of the experience of the face versus color. What is it? That's the question. So just to summarize the clues from previous research, first from face area, FFA, V4, V8, or middle temporal lobe. The neural activity in these areas are strongly, first of all, correlated with, and also necessary for, and can elicit the qualia for face, motion, and the color. Okay, and necessity is established through the specific bilateral lesions of these areas, and then people have reported, loss of face, loss of color, or loss of motion. That sort of suggests that there is something in these areas that generate or corresponds to the qualia of face, motion, and color. And importantly, when stimulated, each of the areas seems to generate these, qualia. And why? That's the question. So to address this question, what I have been proposing, and also many other people also have similar kind of idea, is that we need to understand these kind of situation in the following ways. One is that what is the structure of the qualia, And then another one is what is the causal structures? And then finally, to understand the relationship between these structures. And this week's lecture is dedicated to some extent on what it is that what I mean by each of the structures. Okay, and the first one, Qualia structures, corresponds to, let's say, in this case of listening to some kind of sound, gives rise to experience, and then that experience itself, the relationship or similarity or inclusion or something like that gives rise to this shape of the sound or structure of the sound. And by this, just briefly, in the case of the sound of a C is similar to D or B in the tone. However, when you go farther, different kind of tones, C, D, E, F, and then A, and so on, then one octave above C starts to sound similar. So if you encode the similarity of the sound, then the sound structure has been proposed to look like, helix like this one. C becomes similar to this one. So here, the similarity is encoded in terms of distance in this structure. And a different kind of qualia has different structures. That's sort of the idea here. So what is the causality structures? That's the next topic. So the brain, just to remind you, is a huge network of neurons, and it's really huge, humongously huge, and we can't really have any idea of what kind of structure it is in reality. And the number of the neurons in our head is roughly 10 to the 11 neurons. And in the current Earth, we have roughly 10 to 10 people, 8 billion people. So it's 10 times more neurons in your head than the people on the Earth. That's the level of this gigantic structure in the brain. Okay. And by the way, I might ask you these numbers for this this week's questions. It's pretty easy, so just remember this. Okay, and also this corresponds to that each of the neuron is connected with roughly 10,000 to 10,000 or maybe sometimes 100,000 neurons, other neurons through synapses. Okay. And the key here is that each of this connection is not to directly passing some kind of message or information. Okay, unlike people, each of the neurons doesn't speak to each other. What it does is just to change the probability or the likelihood or tendency for the other neuron to become more likely to be excited or less likely to be excited in the next time step. Okay, so excitation or inhibition of the connection through of the interaction between one neuron to the other, that is the essence of this neural network. And that when it's properly configured, it can compute something, and then that result in many other behaviors or sensory induction or transduction of the signals and things like that. And there is something going on inside the brain of this causality structures or causality network that seems to give rise to this face or motion or color qualia. And in terms of the integrated information theory, this corresponds to what it has been talking as information. But information in IIT is always intrinsic information. And to unpack this a little bit, this is difficult, so you don't need to remember, and I wouldn't ask it in the quiz, but it is the way the network causally constrains its past and the future. If you remember this, figure from the last week, to compute integrated information, the first step of this integrated information started with going from the current state of these neuron pairs, and then how this neuron pairs of state transits into the next future of on, and also where it came from in the previous set. So these are all defined by this, modulation of the excitation and inhibition through the synaptic connections between neuron A and B, and that corresponds to this causality structures. In this case, it's extremely simple, but you can imagine that each of the neurons is connected with thousands of other neurons, and each of the neurons has a unique way to connect to other neurons. Sometimes it's neighbor, sometimes in a long range, and that patterns of the causality has a huge possibility. But it seems that our brain is having this type of the connectivity in a preferential way, in a specific way in each of the brain areas, and that most likely corresponds to, or at least underlies with our specific qualia. Okay, so then what I mean by this content of consciousness. So this is an again a very simple recap of the integrated information theory. Initially, we have this repertoire or patterns that this system can take, and then we compute this how much it constrains itself, intrinsic information, and then how much we lose by if we cut the system minimally. That's the integrated information, small files here. Okay. And then the important thing to realize is that you can do this kind of operation, not only the neuron between A and B, but if there is a neurons C, then you can compute that between A and B, A and C, B and C, and also A, B, C as a whole. So you can extend this kind of analysis on all of the subsets of the things that are composed with complex. By the way, the complex has to be determined first to get which one is boundary of consciousness, right? And then within this boundary of consciousness, all of the possible subset has to be computed and then constitutes the patterns of the causality structures according to IIT. And this is very difficult to do in practice to compute, but that's the sort of theoretical prediction. So in the real brain, this is difficult to do, or at least we cannot do that at all in the current technology or current our knowledge. But what we can do is to do the simulation. So Hong and Tononi last year published this paper where they connected these 8 neurons, A, B, C, D, E, F, G, H, in this way. And then each of these neurons are neighborhood, between the each, like C is connected to both B and D, and the D is connected to C and D, and so on. It constitutes this one line of this network. And each of these neuron is also, excites itself. And then when you set this, weighting parameter or excitation inhibition parameter to some extent, then you can actually compute this integrated information for these sets. And even just 8 neurons, remember we have 10 to the 8 neurons in the brain, the number of the possible ways to consider the subset is really humongous. So this part is the all the possible combinations of A, B, C, D, E, F, G, H, one at a time. And then the next one, this is all the pairs, A, B, B, C, C, D, D, E, and so on, but only those pairs that have a non-zero integrated information. And in this case, let's say, H and E is not connected directly at all. So in the next time step, it doesn't influence its future. And one step before, their E and H state doesn't do anything in the jointly on either E or H or both EH, and therefore they do not give rise to any kind of phi. So that's a phi of 0. So here, the two neuron pair only happens for the this simulated network only with the neighbors. And then this is a all 3 neuron network that has a non-zero 5, and then 4, 5, 6, and so on. Okay, and then this is already super complex, but what they did was to also reveal how they are connected or how they are related to each other, and that's impressively, visualized by this super complex figures. And you don't need to understand this, and I wouldn't ask this in the quiz. But what they did was to visualize the sort of the magnitude of this small phi, or how this small integrated information is related by the some kind of encoding of the magnitude and how it's related to each other. Okay, so let's say, for example, ABCD here is related or closely located to ABC, or CDE, or BCDE, because it's kind of similar to each other, connected, and then it's far from other things like D, E, F, G, and so on. So this embeds the sort of the structure of the causality among this 8 neuron system, and you don't need to understand this complex things. And in general, this week's topic is highly advanced, so you don't need to understand everything I'm going to say. Okay, so in the simulated brain, we can compute the integrated information to some extent, but how can we actually make a progress? So what one of your tutors, Angus, has been working on is to apply this type of the analysis on recording obtained from the flies. So the very small Drosophila flies actually have neurons inside of their brain, and the number of the neurons they have is 10 to the 5 or something like that, and 10 to the 5 to 10 to the 6. So it's much smaller compared to the human brain, but it's again super complicated. Each of the neuron is connected with others, and the structure of this brain is, globally speaking, it's kind of similar to the mammal brain like structures, how the sensory information is connected and collected into the central brain, and then it's it's used for the future control of motor behavior and so on. And what we did was to use these signals called the local field potential. It's similar to EEG in the human brain, and then try to generate this integrated information structure out of this. And this is just a sample kind of integrated information structure that Angus got. So here, this is a sort of the we can only compute integrated information structure or causality structures up to recording from 4 electrodes. And so what we did here is that, let's say, naming it as A, B, C, D, we computed how these the 4 channels constitute this integrated information structure. These yellow dots are the ones that are the contribution to this entire big phi, entire system level integrated information by each of these electrodes, and then green ones are the ones that are the pairs of these neurons or channels, and then the blue ones are trios. Let's say this one is D and B and C. So this is a DBC channel correspondence. And purple one is the ABCD channels. And skipping all these details, what we found is that in general, the recording from wakeful flies are much more bigger in terms of structure, in terms of the magnitude, and also more complex compared to the anesthetized flies. So that's in a sense consistent with what IIT has been predicting about the biological system, and so this is something nice that we kind of working on these days. And previously, we also worked on whether this kind of thing can be also computed for the humans, and it's much more larger scale kind of recordings, intracranial recording compared to the flies local field potential, but still there we are able to get the perceptual report from subjects and then computed integrated information structure. And there what we did was to show some kind of stimulus such as face or blank or something like that, and then we masked it with input to the other eye. This is a continuous flash suppression situation, and then subject reported whether it was a face or not, and then rated the visibility. And then we then focused on these recording areas of the face area, as which is supposedly we didn't do that, but if we were to stimulate these areas, that would probably have elicited face percept in this epilepsy patients. And then we did the analysis, and then looking at all the possible subsets of the 4 channels, 2 channels, and 3 channels, and so on, like Angus did. And here is the results of this paper. So what I'm showing here is 3 different structures that we computed. The left side is this face percept that was elicited during the backward masking experiment. So here we showed some stimulus, face stimulus, and then masked it with a noise stimuli just after that. And then here is a similar kind of experiment, but using the continuous flash suppression. And then the right side is the case where we made the face invisible by the continuous flash suppression. So the right 2 sides are the cases where we ask the subject to do the same task, and then the left side is completely different task. But in terms of the quality of the experience, these left 2 sides correspond to seeing a face, and the right side corresponds to not seeing the face. And then what we found here is that structure follows basically the percept rather than the tasks or stimulus input itself. Okay, so we are actively working on this kind of experiment and also data analysis to see the relationship between the causal structures and the qualia, and that can be investigated empirically. So the question now we can move is that causality structures related to qualia, and then are they unique in FFA and B4, B8 and MT to generate all these face, color, and motion qualia. The idea is that if we were able to compute these causality structures at the level of the possibly single neurons or maybe population of neurons, then maybe there is some kind of a level of the analysis which really uniquely determines the shape of this integrated information structure or causality structures that are unique to face on one hand, and then unique to color, and then unique to motion, and all these structures or shapes are similar across all the people who can experience all these things. And once we get this structure, maybe we can also reverse infer that when you observe this shape in this particular area, then this person must be experiencing motion or color or face. And that could be done both in a wake state or a dream state or drug hallucinatory state, or with or without reports or with or without attention. It doesn't matter. That it could be the direct relationship between the causality structures to the qualia. That's the hypothesis, but we don't know whether it's going to be true or not. And then to address these kind of questions, again, this is a super advanced topic, but I'm not personally doing this type of experiment, but the recording technique has been improving really massively over the last decades really. If you remember that by the time that consciousness research actually started, it was around this time, 1990s. And there, we had human neuroimaging started, and then started to see monkey binocular rivalry experiments and all these exciting kind of time. However, as you will see that after that, the number of the neurons that people were able to record at the same time, which is extremely important to measure this causality structure, steadily increased in a log axis. Initially it was like only one neuron at the time, where we can have no idea about causality structures, up to 10, and then 100. That's the sort of 2010. And then recently, this neural pixel probes has been invented, and then now we can actually record up to 1,000 neurons. And this is a brain of the mouse, where this neural pixel probes were penetrated in the brain in many areas. And in Monash, we also do this kind of recording in mice and rats and also monkeys. And then what it can do is that with a single electrode here, they can record 70 neurons in the visual cortex, 60 neurons in the hippocampus, and thalamus, 200 neurons at the same time. And these analysis, these data recording technique, together with the computational analysis development, will allow us to get to the sort of the causality structure analysis into the future. And that's a pretty hopeful kind of blueprint into the future. So in summary, this first part, which was quite advanced, and so you don't need to understand everything I said, but just look at the key figures and also remember the key numbers, like number of the neurons, the number of the synapses. Okay, integrated information can be computed in a hierarchical manner. So from A, B, C, we can compute A, B, A, C, and C, A. And that's a sort of a hierarchical manner or composable manner that I'm talking about. And then we can estimate such a structure of the neural network in principle. And then the hypothesis is that, or at least it's not really a hypothesis. Okay, so when there is this specific excitation and the inhibition and the connectivity structures, then this causality structure should depend on the specific neural connectivity. And this causality structures has to be specific within an area as well as across the areas, to the extent that neural connectivity patterns are different between these areas. And these are most likely to be the key to explain why faces, colors and emotions are likely to be generated in different areas of the brain, and also at the same time experienced in us at the same time. And then the question is that can causality structures explain a different qualia? And that's the topic I'm going to explain in the next part. Okay, so next I'm going to talk about how to characterize these causality structures and qualia. And one thing it becomes clear when you start to think about this investigation in the way that I propose is that we don't actually know much about these qualia structures actually. And that's the way, that's the next thing that I'm going to talk about. So for example, just think about, seeing this in a Mona Lisa face. Presumably your experience of the face is, the composition of the eyes, left eye and the right eye, and also nose and also mouth. But it's not the kind of, thing that, you only experience this, entire face, or for that matter, everything is kind of visible. And then each of them is actually also composed of many different kind of subsets of the parts. And then eventually, you also are experiencing all these, each of the parts as itself. And this is related to, in a sense, the compositionality of the experience as IIT is proposing. And that was a case of the face. And I already, pointed out that sound may have a different kind of a structure, such as this, helical structure. And the color qualia also has a different kind of, qualia structure. So seeing red, for example, at this point, implicitly at the same time implies that, that point that here is similar to orange or yellow and the purple compared to the, green. And this counterfactual, not reality, or sort of the hypothesized similarity is one of the most likely feature of the color qualia structures. And it becomes more striking once you notice several things, actually. One is that at each point in space and time, two color qualia cannot be actually experienced at the same time. And this is probably similar to the structure of taste or smell or touch, where two things presented at the same time, same location, merges, and then to generate different kind of qualia. And this can be a bit different from other types of the qualia, such as visual or auditory types of the qualia, or such as, a visual motion. Let's say, you can actually see something moving to the left and also to the, up or something like that at the same location at the same time, right? As well as faces also can be transparently presented, and they see two faces at the same time, same location. And the sound is also the case, same. There may be some kind of interference when it's close, or let's say in the case of a motion, when the visual motion is in the same similar direction, it merges as well. But in the case of the color, it seems it's always a case that you can't just merge any two colors. And also another type of the structural property of the color is that we can't lose color qualia as its own. So for example, what I'm saying is that you cannot lose, even if you have a very specific brain lesion, you can't just lose color of red. I talked about in a week 4 or 5 where introduce this brain lesion called achromatopsia. And these patients who report the loss of color, or upon examination, they are revealed that they don't have a normal color sensation in one particular area. Let's say the top right quadrant and so on. They can lose color sensation, but it's always loss of color entirety. From not only the red but also purple, blue, yellow, green. Everything gets lost in one particular area of the visual field. This is not that immediately clear why it has to be the case, but at least so far in the clinical report, we have never encountered any person who reported one particular color experience, but not others. Okay, so and maybe this is also the case for the visual motion or face and sound and so on, but this family of this similar qualia to be lost at the same time may be related to neural circuitry that generates each of the qualia. We don't know that very well at the moment. But there is some kind of implication for neural circuitry that underlies these qualia and the causality structures possibly. So far, this has been possibly pretty abstract, and so I'll just try to make it more concrete. And we may I may ask this in a quiz. So how can we examine and characterize qualia structures? So in the classical psychophysics, what we tend to ask, especially in relation to consciousness, is that did you see A or not? And then you say yes or no, and then with some confidence rating or something like that. And that's been the prototypical experiment that we have examined quite in detail, quite a bit in detail for the blind side experiment, for example. And then the proposed Qualia structures like physics would be not to ask about, did you see X or Y, but rather, to make it broad, what did you see? And also, how did you see it? And then these kind of question has much more freedom and may need to, adopt the response and the analysis strategy quite a bit. So the first one, what did you see, would require verbal report or typing report, and how did you see would require similarity judgment or inclusion, or is A included in B or something like that, and other types of relations needs to be investigated. So in an example of the what did you see kind of experiment, first let's consider this classic paradigm of the sparring paradigm. If you are shown briefly this, by the way, this is highly related to Qianshen's initial presentation example in the week 1. If you are shown this type of the letter array for just 50 milliseconds, and then you tend to have an impression that, oh, I saw 12 letters. But if you are asked by the experiment, which letter did you see, or did you see A, or did you see O, or did you see W, and so on, it turns out that people can report only up to 4 letters correctly. On the other hand, this what sparring actually showed was, after showing this stimulus without a mask, if there was an auditory cue to direct your attention to either top row or bottom row or middle row, then that can recover again 4 letters. So that he suggested this post-cue paradigm, that maybe we are actually initially encoding information for all of these 12 letters and then quickly forgets and so on. But the important thing for consciousness research is that this type of the experiment has been usually interpreted as that the amount of the information in consciousness is up to 40 bits per second. And this has been reviewed by this Noretrandas in 91. Many different kind of the variants of the experiments almost always result in this really lower bound of the information storage. And this doesn't seem to correspond to what we are usually feeling as the rich experience we have across vision and also audition and touch and so on. And maybe there is something wrong about this conclusion, but why is it? That's the question. And my proposal, or our lab's proposal, is that if you ask, did you see X or Y or Z, from the experimental point of view, then you can't really capture the richness of the experience. But if you ask, what did you see, then it's much more proper to address this kind of question. So to address this kind of thing, what we came up with is this new paradigm, experimental paradigm, that we call massive report paradigm. So here, what we show is something like this type of brief presentation of the images, and then we ask people to report what they saw in various different ways. Okay, and this is project owner's project Cao Kou last year, and we are now writing up the paper for this. And in this particular case, we asked the subjects to report 5 words that they saw after each of the image. We didn't show multiple images at the same time in this particular experiment, and then also ask the confidence rating. The quite different kind of design from the previous experiment is that we use lots of images, and we use it in a way that subjects cannot expect what to see. And this is impossible to do with the previous paradigm, because previous paradigm we wanted to have an answer and also foil beforehand, but we basically completely discarded that kind of concept. And then we asked people to type in the report or what they see, and then we use the online experiment platform and to get lots of participants. And then lots of participants, which was not possible to do with the traditional paradigm, allows us to also get the statistics and also reliability of all these responses. And then Kianshen, another TA for this class, is doing this kind of experiment, but using not the words of a free report, but showing these small patch of the images at the time, and then asking them to report whether they saw it, so that a particular patch at the location they are specified. So this is closer to the classic paradigm, but it's much more radical in the sense that we here show one of the images that is never possible to expect, and then we show one patch and then ask, did you see this or not? And then after that we show another patch, did you see or not? And then another patch, and then did you see or not? So this form of the image-based massive report paradigm can be also done to address the same questions. And then as a result, or sort of the preview of this result, which we will publish in soon, generally speaking, the massive report paradigms reveal the structure of experience that matches with our usual sort of the intuition and also feeling. And any moment of experience seems highly structured, as IIT also proposes as a composition, information integration. And also people can differentiate many different concepts and images without much difficulty and a very short duration. And then another example of to go into the Qualia structure is to use this similarity ratings. And here, what Ariel did, I think I introduced his experiment in very early in the lectures. He showed a patch of the color, let's say blue, green versus red, or blue versus pale blue, one at a time, and then asked them to rate the similarity. So here, let's say this is the case: blue paired with pale blue. People may say that it's quite similar, but not the same. So it's a gray scale here. On the other hand, when the red is paired with the green here, most people say that, oh, that's a very different color, so it's black. And then if you pair all the possible, colors, then you can generate this similarity matrix. And then from this, we can deduce some sort of the color structure, experience structure of the color. And this color structure experiment can be used to address the question of the, do we see color in the periphery? We spent a lot of time in week 4 and 5 about this question. If you remember, in the standard psychology textbook or physiology, what you learn, relatively early in the visual systems is that the cones that are supposed to be responsible for color experience decrease its density from the fovea, where zero eccentricities falls off really quickly. And as I said before, I might ask again in the quiz that fingernail from at the arm's distance is roughly 1 degree or 1 to 2 degrees. And so outside of this, let's say 10 degree is almost like the edge of the or halfway of the display at the arm's length of the computer. And around there, you almost see that the cone density is extremely low compared to the fovea here. It's around 100, 150, and here it's like less than 10. And based on this, many people and the textbook possibly say that we actually do not see color as vivid as in the fovea. Okay, that has been the sort of the claim before. But I argue in the lecture that, these people who claim this do not usually consider about the, magnification of this, effects in terms of the cortical magnification and axon projections. And so if we magnify the stimulus, like the right side, then there is actually no loss of color vividness into the periphery. And that explains why we experience the blue sky in the periphery all the time if you go outside, because, blue skies are, by definition, it's blue in a uniform way, and that effectively stimulates lots of, peripheral retinal stimulus, retinal cones, and that generates this blue experience. But which one is true? That's the question. You might, I may or may not have gained your, so trust over the last weeks, but some of you guys may doubt what I say, and some of you may, believe in what I say. And the nice thing about science is that we can actually test this empirically. And what I really did was to use this display settings, and then to compare these similarity structures between phobia pairs and periphery pairs, as well as the phobia peripheral pairs. So here, I expect that phobia stimulus should look very different because of the lots of cones, but periphery to periphery may become much more sort of blurry, and we don't see it well. And then further on, central to the periphery becomes even kind of weird, because central is very vivid, and the periphery is very vague. And that's what we tested. And then what we found is the following. So this is first of all, the demonstration of this task. So after you fix it at the center, and in the real experiment, the speed of this display is much faster. But what you do is something like this. So at an unpredicted location in each trial, you are given two stimulus, and then you just make a comparison of the similarity across all these trials. Okay, and then what we found is, on average, something like this. What it means is that in the end, central and the peripheral combination is almost like identical in terms of similarity structure. So this central combination is very similar to peripheral combination, and the periphery to central combination is also very similar to central combination. And it always seems to show this kind of ring structure. As I mentioned, the red is closer to the orange and the purple, pink, and very different from green. And this kind of structure seems to be preserved across the visual field. And there are important, individual differences. For example, there are, by the way, this is the example of the 15 subjects out of 100 subjects that we have tested so far. Online, that phobia to periphery always is a gradual. Some people like this, and then this, and then all these things. All these people seems to gradually rate this similarity. However, there are some people who are very strict that only very neighbor color is similar, but not any further colors. And these another type of the person says that there is some kind of clusters of the categories that are considered identical but not others. And why this kind of individual difference arises is not really clear at the moment, but we are trying to get to the bottom of this. So in summary, in this second part, I try to explain that the Qualia structures can be examined by addressing what do we see, using a maximal report paradigm, or how do we see using a similarity rating paradigm. And both of them, these Qualia structure psychophysics, are relatively new and minor within the consciousness research. And most of the research uses still like, did you see X, or did you see X or Y type paradigms? And they are, in their own very important and useful. But to get to the question of the structures of Qualia, that's probably not really rich enough. And as we are developing this, we encountered various problems. So, paradigms and also tools for the analysis need to be developed. And once we have these Qualia structures together with the causality structures that I mentioned, introduced in the first part, then finally we can start to address the question of how we can actually test the correspondence or relationship between the Qualia structures and causality structures. And that's going to be the next video. So in the last part, I'll talk about this last part of this arrows between the Qualia structures, such as sound or face or motion or color, and this proposed integrated information structure or causality structures. How do we manipulate or examine this, related relationship? Okay, so here's the exemplar face Qualia and also compositionality. And most likely these are generated or related at least to the face form face areas neurons. And idea here is that integrated information structures generated by this, 4 neurons or 4 population of neurons or whatever, we still don't know. There is probably some kind of combination of the integrated information, and they are potentially corresponding to this structure of the face experience. To any kind of experiment is always necessary to test the hypothesis, and one of the promising paradigm, I think, is this rewiring experiment. This is a quite radical kind of experiment, but it has various interesting properties. So what we did, what the Marian Clutter in MIT did roughly 20 years ago, is that in a normal brain, the eyes or retina actually extends thalamus, LGN, and also speria colliculus, as we discuss in the context of the blind side patient. And then from LGN in particular, that axons goes into the visual cortex. That's a sort of the normal visual system development. On the other hand, the auditory system that we haven't talked much has a completely different kind of structure, and then ear, then the cochlear extends the axons into brain stems and so on, and then eventually goes to this inferior colliculus, and then that projects the axons into MGN, medial geniculate nucleus, of other part of the thalamus, basically. So the lateral and the medial geniculate is actually close in the distance. And then from MGN, axons goes into auditory cortex, and then that generates this, auditory cortical areas. And then what they did is, in this rewired animal, they cut the connection to the speria colliculus first, and also they induce some kind of branching from normally going to speria colliculus into MGN. And then that resulted in this rewired animal whose axons goes from MGN to auditory cortex, which are actually responsive to visual stimuli. So here, they also cut the connection from the auditory cortex in auditory apparatus, like a cochlear, to MGN. So auditory cortex in this case developed like an auditory cortex. And this was done mainly in ferrets, and then they performed also the various interesting kind of the behavioral experiments. So in terms of the cortical physiology, what they found in this Nature paper is that within this visual cortex of the ferrets, the V1 neurons generate standard cortical map and also orientation preference and so on. And this is a very standard kind of map preserved across animals. And then each of the neuron in V1 located here in the soma, the neuron body extends axons and also dendrites around its neighborhood, but also it extends to somewhere far away. And this kind of neighbor plus far away kind of connection is very important for building this lateral connection of the visual cortex. And that's the kind of thing that model neuron system that the Hon and Tononi kind of simulated in this 8 neuron line experiment over there. But leaving aside, this critical thing is that the rewired A1 cortex. So rewired A1, now this ferret actually received the visual input, and then that gets projected into the auditory cortex. And then there, the auditory cortical neurons also demonstrate when they use the optical imaging, cortical map that looks like, but it's deformed in the visual systems, okay? And then compared to the neuronal projection of connection pattern in the standard normal A1 neurons, which is only like, highly densely connected to neighbor only, rewired A1 neurons show, here's a cell, cell body. It has this extended kind of, dendrite pattern, which is very similar to V1, visual cortex. So this means that local neural connectivity and the structures and the potential activation pattern itself of the auditory neurons now became similar to visual systems, but it's not quite identical, okay? Then what happened, or what they did in an ingenious experiment, in von Melchier's experiment. By the way, this paper is 2 papers and published in the same issue of the Nature, which is just amazing. And it happened only a few times, I know, in the my history of the science. But anyway, so what they did in this behavioral part of the experiment is that to put the ferret into this part and then presented they either light pattern onto the right side or left side. And then also sometimes they also presented the auditory stimulus through this speaker. And then they trained the ferret using this normally developed hemisphere where no rewiring has been operated. They trained the ferret to go into here when they hear auditory sound, and then go to the right side of this reward spot when they see light, okay? So if it's vision, go to the right. If it's audition, then go to the left. And then ferret learn this very quickly. It's very easy discrimination. And then the critical question they asked was, what happens for the first time when they present the stimulus to the eye of the rewired brain? So now the visual stimulus goes through the eye, and then that gets projected into auditory cortex. The other side of the auditory cortex is that standard auditory cortex, and the visual cortex doesn't receive the input directly from there. But what happened was that the ferret actually reported, seeing here. So now the rewired auditory cortex seems to, at least, be consistent with the hypothesis that this, based on this changed connectivity and also causality structures, now they can generate a visual experience, and that's what the ferret reported. And I'm saying not only this particular experiment, but also this kind of experiment or application has been done in humans where the experience is restored or changed by the rewiring or sensory substitution. In these kind of situations, newly extended or acquired connections, as long as they seem to be similar to the original sensory experiential apparatus, it can generate a qualia of the original modality, even if you are using auditory cortex or visual cortex or somatosensory cortex. So in summary, once qualia structures and causality structures are obtained, in the future possibly, we need a mathematical theory to test their correspondence. And I'm not talking about this particular part, but in other papers, if you're interested, I'm talking about this kind of stuff, category theory. But the important thing for this lecture is that to understand this structural relationship between qualia structures and the causality structures, there are many empirical paradigms that can be used. Rewiring paradigm is one that I briefly talked to today. But individual differences and also synesthesia, congenital blind, and many other things can be actually used to test the structures or structural correspondence between qualia and causalities. And this is a highly promising direction of the future experiment. Okay, so concluding the remarks. First of all, congratulations to all of you to finish this course. And this week's quiz is going to ask again, like last 2 weeks, it's going to be easy. And so don't worry about it too much. And the last part, I'm going to briefly give you a sort of the overview of societal impacts of this, type of the consciousness research. First of all, so the consciousness research, as you might be able to imagine, has a direct influence on the clinical implication. Sensory substitution or restoring qualia through brain-machine interfaces, as I mentioned, in the last section, is active area of the research. And also consciousness meter, like we talked last week, perturbational computation, perturbational complexity index is another active area of the research. The photo that I'm showing here is a boy who was hit by the car and then went into a coma. And the parents and doctors actually thought that this boy is never possible to recover. So all of them kind of signed the contract for the organ donations. But this particular boy actually recovered his consciousness just 1 or 3 days before the operation. And since then, he is now completely recovered and then playing basketball and things like that. But at this moment, we still don't have a good way to predict the recovery of consciousness after the traumatic brain injury. And these types of the clinical application is requiring definitely the understanding of how the consciousness is supported by the neural activity. Another things, the direction academic curiosity types of the question is, the mind-matter relationships or whether the laws of physics are enough to explain consciousness, but also development and the evolutionary aspect of the consciousness is also quite an important topic. Like this newborn babies are going to increase, preterm babies are going to increase due to the medical, advancement. And also we are increasingly, aware that many animals outside of the human or, a distant kind of, phylogeny, like octopus seems to show highly intelligent behaviors. And also flies and also bees also show some evidence of cognitive capacity. And whether they are conscious or not, and if so, how similar their consciousness is, or how different they are, can be a very important questions for the animal ethics. And speaking of animal ethics, this engineering technique, bioengineering technique called organoid is also becoming a quite important issue. So right now, we are now able to keep the brain of the pig alive, putting the brain itself outside of the pig's body for 2 weeks or more. And also we can develop this organoid, which is coming out of the stem cells and then develop the brain-like structure on its own without a body or a blood vessel or something like that. That part we will artificially kind of supply. And then this starts to become like, philosophical thought experiments that people have thought for a long time. When the brain is floated onto the vat, do they actually experience consciousness? And if so, can we actually generate such things, or can we do this kind of experiment ethically? This is a kind of a question also again related to, or at least attached to the consciousness research. And also finally, this is a figure from science showing that there is an already futuristic kind of era that robots are substituting some of the human labors, and the more that inside of the mechanics of this robotics becomes intelligent using artificial intelligence, things, then they might start to speak like humans, and it may feel like you may feel this entity as more conscious than non-speaking patients and so on. And then in this case, what do we do in terms of again ethics, or what we can do with this for the engineering or commercial purpose and so on becomes important. And the brain-machine interface is also possible to move these robotic stuff through your recording of the brain and so on. And how much of what we can read out of the brain, consciously or non-consciously, is also another matter of the debate. And to enable all this consciousness research, we do need an interdisciplinary research. And sometimes you may heard that we need an interdisciplinary research, but in the case of the consciousness, it is not just like a nominal or phrase, but it really requires an interdisciplinary research and collaboration. And I myself have some kind of background in neuroscience, but did the psychology and also read the philosophy and collaborate with these people. But also through this interest in consciousness, I also learn and also collaborate with the people who are studying this evolution, cognition, language development, pharmacology, and clinical science, and then biology, and also to make it applicable into engineering. Collaboration with the engineer, computer scientists, and also physicists, mathematicians, artificial intelligence, robotics, and information theory is becoming more and more important. So into the future, you may see that consciousness research extending into different kind of fields, and also if you are working not in the consciousness research right now, but in these surrounding areas, maybe we'll have potential collaboration into the future. So the concluding remarks. I first wanted to say that the views I provided throughout the course are highly simplified, and I realized that I probably misled some of you guys, and also gave you biased information. But you do as a sort of the introduction to the consciousness research, I thought that this kind of bias is never avoided. So I just went ahead and then finished it. But I just wanted to say that there are a lot more to learn, and also there are many things that can be told, and you do need to go from here by your own to develop further understanding. And also, I used to say this at the beginning of the lecture until the last year, but this year I tried not to say that. But almost everything in this course material is likely to be revised in the future. And this is not a bad thing, but it is a more sort of the property of the science itself. Even, my previous mentor, Joe Bogan, who did the split brain experiment in the since 60s to 70s and so on, he said that except for anatomy and mathematics, all he learned when he was in a university was changed, and before he died. So he strongly recommended to learn mathematics and anatomy. And I would say that even anatomy and mathematics also changes because science improves, including mathematics and so on. And as I mentioned, the category theory that I'm learning over the years seems to suggest that there has been definitely progress and also change, revision even in mathematics. Similar things definitely happens for the psychology, neuroscience, and also information theory and so on. So this is a rather exciting thing that you are going to find out into the future. And finally, consciousness is so fundamental that people may not even notice how mysterious its relationship with the brains. And when I give this kind of talk to lay people, it's quite interesting, but some people never get why the question of brain and qualia are mysterious. It's if things are so fundamental, then some people just can't get why this is a problem. But hopefully, through this course, I hope that most of you get why this is so weird. And as such, due to this nature of the consciousness as a topic, it would be good, material for you as a psychology student to, improve your skills to be critical and also empirical and also quantitative and logical about these facts. Don't take, initial, your impression as it is, but also don't also, trust whatever, other authorities said, including myself, but you can actually empirically test your own experience and then, think about why, you are experiencing the things in the way that you do. And that's the kind of, attitude that is very important for your future, regardless of whether you are in academic or outside of field. And I hope that you enjoy the class and good luck with your future. Thanks for listening, and hopefully I'll see you again. And I just want to thank the members and also the collaborators in the past and current and future for making this possible up to now. All right, see you until the next time.